Hi everyone. Today we're going to be talking about the history of DNA sequencing as well as some of the more common methods of sequencing DNA. So what is DNA sequencing? Like the name suggests, it's basically having some sort of program and technology to read out the bases of a given strand of DNA. And there's several real-world applications to this. So one is biosystematics. The definition of a species has varied a lot over time, and DNA seems to be one of the strong candidates on how to split up certain clades in species. And so DNA sequencing research has a large role in biosystematics. And another, obviously, biomedical research benefits a lot from DNA, being able to see the genes and proteins involved in certain diseases and certain crucial functions of the human body, as well as forensics and its applications there. So we'll also go through a brief history of DNA sequencing from the early 1970s to the present day. And here are just some special projects. As many of you know, the Human Genome Project, which was in the early 90s and early 2000s, sequencing the first complete human genome, as well as the UK 10K Genome Project, which was sequencing 10K British people in the 2010s, and as well as the B10K project, which was sequencing 10,000 species of birds. So the first sequencing method we'll be talking about is Sanger sequencing. This was the first common sequencing method developed by Frederick Sanger in the early 1970s, and it's also referred to as first-generation sequencing. The way it sequences DNA is through the chain termination method, and this method requires PCR, which is polymerase chain reaction, that basically amplifies the DNA that wants to be sequenced. It requires ddNTPs, so dideoxynucleotide triphosphates, as chain terminating bases, as well as gel electrophoresis in the end to determine the sequence. So while Sanger sequencing was the first method that was developed, it's really time consuming because you have to clone the DNA many times, as well as use the chain termination method, which is inefficient, and it costs more per nucleotide than other methods. One advantage, though, is that it is able to do longer reads up to 1,000 base pairs with pretty good accuracy above 99%. So how is Sanger sequencing accomplished? So as we talked about, the first step is PCR, and this is done with fluorescent ddNTPs. So PCR amplifies the target DNA, and as it's amplifying, some ddNTPs will be in the solution as well as normal dNTPs. And when ddNTPs are sequenced onto the target frame, it terminates the chain and basically stops the amplification. So this occurs at every position along the target DNA strand until we have many DNA strands that are of various lengths across the entire target DNA strand. And in the second step, we separate the sizes of the various DNA strands into a column, and this is done through gel electrophoresis, which separates the DNA strands by mass. And in the third step, we excite the gel electrophoresis column, and that can tell us what the sequence is. So each base will have a different color when excited, and when we line up the wells, as we can see over here, we get the final sequence. So the next type of method is next-gen sequencing, which is also called second-generation sequencing. This umbrella term basically covers a lot of sequencing techniques developed in the early 2000s and the late 90s. But the main one we'll be focusing on is Illumina sequencing. And this does sequencing by sequencing through synthesis. And this yields short reads, but with extremely high accuracy, and it also has lower cost and less time. And up to today, Illumina sequencing is the most commonly used sequencing method. So Illumina sequencing is performed on a flow cell plate where a lot of DNA strands are housed and sequenced at the same time. It starts with bookending the target DNA strands. This allows it to attach to the fuel cell plate, and their bridge amplification occurs where the DNA is amplified by using DNA polymerase on the flow cell plate. From there, we have a lot of strands on a single flow cell plate, and then it can then be sequenced by synthesis. And what this means is that we have fluorescent nucleotides, and when they're sequenced, they give off a fluorescent light, and this can then be picked up by the sequencing machine all along the flow cell plate, and we can sequence a lot of DNA strands at the same time through synthesis. And the third and most recent sequencing method we'll be talking about is PacBio Sequencing, or single molecule real-time sequencing. This is also known as third generation sequencing, and it was developed in the 2010s and is the most advanced sequencing method today. So a distinction from other sequencing methods, PacBio sequencing is capable of extremely long reads. So we're talking up to 100,000 base pairs per read, and also with really high accuracy. So it's good to note that sequencing methods like Illumina prefer shorter reads because longer reads will cause increasing disparities in accuracy. But through a technology called circular consensus sequencing, PacBio is able to sequence extremely long reads at high accuracy. This is done through the use of circular bookends on the DNA strands. So if you look at the diagram here, we have a single polymerase able to circle around the DNA strand many times and sequence it continually. So while one read may be inaccurate, the DNA polymerase is able to continually sequence many reads, and then those can be compared to determine the consensus read, and this yields really high accuracy. PacBio sequencing is able to do this because each DNA strand is in a well called a zero mode waveguide, and there's a DNA polymerase attached to the bottom of each one and a photoreceptor camera, or light I should say, at the bottom of each ZMW. And also this can perform sequencing real time and read the data. So this is just a brief comparison of the sequencing techniques. As we said before, Sanger sequencing was the first method, followed by next-gen and Illumina sequencing, and more recently SMRT sequencing or PacBio sequencing. Each method has its advantages and disadvantages, with PacBio sequencing able to read really long reads and also at very good accuracy with circular consensus sequencing. And also Illumina sequencing providing short reads but really high data volume at a really low price. Sanger sequencing has really high accuracy but is not used a lot due to its high price compared to other modern sequencing methods. So this was just a brief overview on several DNA sequencing methods, and I hope you found it helpful.